membrane proteins serve a variety of functions. Some of them are transporters. They act to move substances in or out of the cell. And we'll talk about several classes of those transporters and differences in the way they act. Enzymes can be in the membrane, and those enzymes can catalyze numerous biochemical reactions, including biochemical reactions that involve the transduction of signals from the extracellular environment. And we'll talk more about that when we get to cell signaling. Receptors can bind to molecules and allow the cell to communicate with signals that come in from other cells or from other tissues. As we've mentioned, there can be cell surface identity markers, usually glycoproteins, that are proteins that have glucose attached to them. There are cell adhesion molecules that are membrane proteins, and those allow those proteins bind to proteins either in the extracellular matrix or on other cells to bind those cells tightly together based on their membrane proteins that adhere to one another. And membrane proteins internally in the intracellular environment, they can attach to the cytoskeleton, and we've talked about that. They can communicate to the cytoskeleton from the outside of the cell. So here's some examples. Here's a transporter. In this case, we have an ion channel that would let ions flow from outside the cell to inside the cell. Here's an enzyme, which is membrane bound, an integral membrane protein that we schematically illustrate as taking a reactant and producing a product. Cell surface receptor would receive a signal from the extracellular environment and cause the transduction of that signal into an appropriate cellular response through mechanisms that we'll talk about. It doesn't look like a radio wave like it does here, but we'll talk about the mechanisms of signal transduction later on. Here's a glycoprotein, glucose attached to the protein, extracellular space. This is an identity marker. Cell adhesion molecules are binding two cells together by the interaction of proteins that have high affinity for each other. And then here is an example of an integral membrane protein that is attached to a peripheral protein that is bound to cytoskeletal elements, in this case, actin microfilaments. And therefore, the shape of the cell can be affected by signals coming from the outside. So diverse functions of the membrane proteins are related to their structures. And we'll see this again and again, that structure arises from form. Form yields structures that yield functions. So, but they do have common structural features. Even though there are differences, since they are all in the membrane, they do have certain features in common. And we talked about peripheral proteins that anchor molecules and attach the membrane protein to the surface. So here's an example of an anchoring molecule that is a protein that's anchored to a phospholipid, in this case, a specialized lipid. So integral membrane proteins span the lipid bilayer. They're transmembrane proteins, and they have nonpolar regions that are embedded in the interior of the bilayer, which makes sense because the interior of the bilayer is hydrophobic, whereas polar regions of the protein poke out from the cell and poke into the cell in the cytoplasm and in the extracellular space. And the transmembrane domain is the part that spans the lipid bilayer, and it usually has hydrophobic amino acids found in alpha helices. So if we look at a schematic of these integral membrane proteins, they often have just they can have just a single alpha helix, which is embedded in the phospholipid bilayer, and the hydrophobic region of the alpha helix is indicated in blue here. That is, the amino acids found in the membrane spanning portion of the alpha helix have the amino acids that are found there have hydrophobic side chains on them, which makes sense because they are mixing with the hydrophobic fatty acid tails of the phospholipid bilayer. And then we can have an extracellular domain and an intracellular domain. Although there are proteins with only one membrane spanning helix, in many cases there are more. And a common motif that is found in a number of receptors of the G protein coupled receptor family that we'll talk about later, just to let you know that we will be talking about that, have 7 membrane spanning helices, all with hydrophobic residues in the portions of the helices that are embedded in the fatty acid portion of the bilayer. Here's an example, a particular example. This is bacteria rhodopsin, which captures light energy, and that light energy is captured by the retinal chromophore, the retinal chromophore, and that capturing of energy pumps protons across the membrane. We don't need to concern ourselves too much with that right now, but this is an example of a 7 transmembrane helical protein. Now, let's consider some classes of membrane proteins and start out with talking about pores. Pores have extensive nonpolar regions that allow them to be embedded in the phospholipid bilayer, and pore proteins, as their name implies, create a pore through the membrane. And many of these pore proteins, or we call them porins sometimes, many of these porins have the structure of a cylinder of beta sheets, a beta sheet that is wrapped around into a barrel shape that creates a cylinder with a hollow center, and we call that structure a beta barrel. And the interior of the beta barrel is polar, and it allows water and small polar molecules to pass through the membrane. So porins, or pores, create a polar channel through which polar molecules can flow in and out of the cell. And here's an example of such a beta barrel pore protein. You can see that the major structure of the protein is one continuous beta sheet that wraps around itself and forms a hollow barrel. And the interior of that barrel has polar side chains that it can allow polar molecules to flow through the pore protein or out through the pore protein. So they provide a gateway for polar molecules to pass in and out of the membrane. That's what pore proteins do. Now let's consider a process before moving on to any particular classes of membrane proteins. We'll be talking ones that are involved in passive transport first. But before we talk about proteins, let's talk about the process of passive transport. We can define passive transport is a movement of molecule through the membrane that does not require any input of energy. And molecules are moving in response to a concentration gradient, moving from high concentration to low concentration. This is a basic chemical law that substances will diffuse from high concentrations to low concentrations, and so that trying to equal out the concentrations, trying to create an equal distribution of the molecule. This lowers the free energy of the system and is thermodynamically favored. And so diffusion accounts for this, or diffusion is the term we use to describe that movement. And as I've said, that thermodynamically that's favorable to create a situation in which the concentration is equal throughout the system. And here's a schematic of that. If you take a colored dye molecules, drop it in water, the dye molecules will diffuse until they are evenly distributed. That is the process of diffusion. That's the process of a molecule moving from an area of high concentration to areas of low concentration until concentrations are equal. Remember that the hydrophobic interior of the phospholipid bilayer repels polar molecules, but nonpolar molecules can pass through the membrane rather easily. So nonpolar molecules will tend to move through the membrane until the concentration is equal on both sides. There is phospholipid bilayers. The membrane provides limited permeability to small polar molecules. If it's small enough, there's limited permeability of those, but very limited permeability to larger polar molecules, as well as fully charged ions. Ions really can't pass through. They're too polar to pass through the phospholipid bilayer. So there's a process of facilitated diffusion. The molecules that for which the membrane is impermeable can move through membrane proteins. And again, they move from higher to lower concentration according to the rules of diffusion. And channel proteins are a class of membrane proteins that create a hydrophilic channel when they are open and allow facilitated diffusion. That is, channel proteins facilitate the diffusion of, in particular, ions, which are fully charged. Ions, when the channel proteins open, the ions will flow through their hydrophilic channel. Another class of proteins that allow for facilitated diffusion of polar molecules are carrier proteins. And carrier proteins, unlike channel proteins, actually bind to the molecules they assist in passing through. So as a result of this, the membrane is selectively permeable to polar molecules, depending upon whether those polar molecules have access to either a channel or carrier proteins that help them pass through the membrane. So let's talk about channel proteins first. Ion channels, channel proteins are primarily ion channels. Sodium, chloride, as examples of cations and anions respectively, can pass through particular channels that are selective for them. So ion channels allow the passage of ions. And gated channels open or close in response to some stimulus. And that stimulus can be an electrical signal or a chemical signal. If the membrane potential, that is the charge distribution across the membrane, changes, that can often cause channel proteins that are gated channels to open up and allow the passage of ions through them. And the conditions that determine the direction of flow of the ion, of course, is first and foremost is the relative concentration on either side of the membrane. So if you have high concentration of sodium outside the membrane, this is the membrane, here's our lipid bilayer here, and you have a high concentration of sodium ions outside the cell and a low concentration of sodium ions inside the cell, and a channel protein opens up and opens a channel that is permeable to sodium. Sodium will flow into the cell down its concentration gradient. And voltage differences across the membrane can affect ion flow, because if the inside of the cell is negative with respect to the outside of the cell, then positive ions will want to flow into the cell, into the region of negative charge. And we've already pointed out that gated channels can be either open or closed, and they open in response to particular stimuli. So here's an example of a channel protein. High concentration of this ion outside, low in. When this channel is open, it will flow down its concentration gradient. Likewise, you could have a higher concentration inside than outside of some ion, and when its channel is open, ions flow out of the cell down its concentration gradient. And we'll continue our discussion of facilitated diffusion by moving on to carrier proteins in the next section.